Welcome to Endometriosis Voices, listening to lived experiences, learning from science. I am Isaac Chebafo, and today we are doing our first research highlight. The decision to review this particular paper as our first research highlight episode was influenced by a Facebook post that was sent to me by someone verifying what the Facebook post was claiming. In that post, the authors were claiming that scientists have found a common bacteria that causes endometriosis, and that treating that bacteria was synonymous to treating endometriosis. This was interesting, if indeed it was true, and I was not surprised to find out that that particular post has been shared close to 4,000 times and has had about 1,800 comments. To verify the extent of that information on social media, I decided to query that search to see if there are other posts of its kind. And it was not surprising that there were several others of its kind, most of which were using pretty much the same title as the one that was sent to me to verify if that was the case. When I became aware of the extent this particular social media post and others of its kind have spread, I decided to dive in to see whether the claim is true, one, and two, whether if it has been made, it's been verified and published in a very reputable journal. The reason why I decided to go down the rabbit hole is that most endometriosis sufferers are experiencing pain that they can't even explain. And so they are looking for something that can, if not treat the disease, deal with the pain. So if they hear that scientists have found this, and then going to a pharmacy to just procure an antibiotic is all you need to cure endometriosis, some of them are likely to just take that information and walk to a pharmacy to try that route. After all, it is an antibiotic and we are used to antibiotic for what it is. But this is harmful in two ways. One, if this is not true, then that endometriosis sufferer who is taking the antibiotics just get all the side effects of the antibiotics, but no reward from it. And then secondly, there is a global public health concern about something we call antibiotic resistance. And the way it works is that if you use antibiotics indiscriminately, especially if you don't complete your dose course or you take the antibiotics when you don't have to take it, then you have some of these pathogenic bacteria develop resistance to the existing antibiotics that we have. And then when someone needs it and you administer it, it tends not to work. As it stands now, a lot of investment is being made into the discovery of the next big antibiotic to deal with antibiotic resistance that is happening. If we are able to slow it down, perhaps some of these funding could be channeled to endometriosis research, and then proper or effective treatment programs could be developed, or the disease could even be understood, and then it will work both ways. So these were the two motivations that pushed me to want to go into the rabbit hole to find out whether this claim that has been made about the Fusobacterium as causing endometriosis is indeed true. So finally I found the paper, and once I found the paper, there were two things that I wanted to verify. And the first one is where the paper is published. And in this case, it's published in Science Translational Medicine, which is a very good and reputable journal. The potential that they had made sure that whatever claim is being made in this document has been verified, or that enough information has been produced beyond reasonable doubt that the claim is not what they saw during their project or study. All those guardrails might have been put in place during the peer review process. Secondly, in scientific communication, there is a particular effort to ensure that whatever is inside your article is communicated in your title. And so I also paid attention to the title. And the second thing is that this Fusobacterium is not just any single bacteria. The Fusobacterium is a genus that has about 13 individual species of bacteria in it. And so when you look at the Facebook post that I shared in the beginning of the episode, you realize that the Fusobacterium was communicated as if it is just one single bacteria that infects you and then it gives you the endometriosis. I kind of had some doubts about whether or not these individuals who authored the various social media posts had actually read the document in its entirety. So when we get down to the specifics of what the paper is about, or what the authors were trying to communicate, they really started making a case for the rationale for their study from the second paragraph, where they spoke about that. Yeah, we understand that endometriosis is a benign case or a benign condition. That means it's not cancerous. But then it has some specific features that makes it sometimes you wonder, why is this disease not even classified as cancer? And one of which is a high level of tissue that we call fibroblast. Usually you'd expect the volume of fibroblast that you see in endometriosis to exist in cancer, but then also there are some specific markers or features that we expect to see in cancer that are not present or available in endometriosis, and so it's still considered as a benign disease. Then finally, they make a case for why the Fusobacterium could be an important bacteria in endometriosis. And they mention that though usually you'd expect the species of the Fusobacterium to be available in the mouth and in the digestive system where they are not harmful or they don't cause any disease, there's evidence that in some cases they have been shown to cause infection in the gums and in the teeth. And then also during cancer, they have been shown to activate some cytokines, which are some chemicals that are used to facilitate inflammation, particularly interleukin 6, which is the IL-6, interleukin 8, and then tumor necrosis factor. This bacteria has the potential to recruit these inflammatory markers to facilitate the occurrence of inflammation. So now they've set up the rationale as to why investigating a possible involvement of this particular bacteria in endometriosis is required. So going into the study, they wanted to find out whether the endometrial stroma from patients with endometriosis contain different types of fibroblasts compared to those without endometriosis, and whether fibroblasts might be associated with the presence of specific pathogenic bacteria. So what they did was that, okay, we know that you are going to find a fibroblast there anyway, but then in endometriosis sufferers and those who do not have endometriosis, do they have different type or different versions of their fibroblast? And then if they do they have any indication that the bacteria could be the one that is either contributing to the overproliferation of the fibroblast or helping the fibroblast to recruit these inflammatory cytokines to facilitate the disease occurrence? This was the question that the researchers sought to find answers to. And then they found out that the transgellin expression is significantly high in the endometrium of the patients who had ovarian endometrioma compared to patients who did not have ovarian endometrioma. Now, the second thing that you'd want to find out is, does the increase in the expression of the transgellin has any role to play in the fibroblast proliferation or expression in there? And so that was the next question that they tried to find out. I'm not going to bore you with the details, but when you read thing here, they said that they found a link between the expression level of the transgellin and the proliferation of the fibroblast. So now they've been able to establish the possible link between the transgellin expression level and then the migration of the fibroblast. Then finally, they were able to establish that high expression of the transgellin also kind of promote the endometrial cell proliferation using an interleukin 6. So now they have established that in patients who have ovarian endometrioma compared to women who do not have endometriosis, the endometrium express high levels of transgellin, and the transgellin promotes the proliferation and migration of fibroblasts, while the transgellin also improves the proliferation or the rate at which the endometrial cells develop by recruiting interleukin 6. So these three connections has already been established. So now let's go back and look at how they sought to prove whether it has anything to do with the Fusobacterium or it does not. The researchers decided to look at first publicly available data, that is research that has been done and published already, whether they could find any evidence. Has already mentioned that there's a possibility that this Fusobacterium could be doing something different in the endometrium of patients or individuals suffering from endometriosis than those who do not. So They did that, and then they said they found evidence that Fusobacterium infection in the vagina is higher in those who have endometriosis than those who do not. And then when it occurs, they affect the endometrium or the way the endometrium behaves. Not just any Fusobacterium that has been linked to capabilities of changing the endometrial microenvironment in endometriosis individuals, but then the particular one that they call Fusobacterium nucleatum. And this infection is not actually in the uterus, but then in the vagina. And when they occur in the vagina, then they have the ability to influence the way the endometrial tissue behaves. And this is well pronounced in endometriosis patients controls. So now we have established the link between transgenic, fibroblast proliferation, and endometrial cell proliferation. And then there's evidence that Fusobacterium infection in the vagina occurs more frequently in endometriosis patients than it does occur in healthy controls. And when they do occur in endometriosis patients, they affect the way the tissue microenvironment of the endometrium behaves. So the next thing that you need to do is ground this in truth. You can't do this in humans. You can't call a bunch of humans and say that, yeah, let me give you money and give you some vaginal infection and see if you develop endometriosis. Nobody's going to give you ethical clearance for that study. And even if you get clearance by whatever means you use, I don't think anybody's going to sign up for that kind of study. So you need to step down and come and do the verification in animal models. So all these other verification that you are doing, whether the transgenic has any role to play in fibrocyst differentiation and proliferation, and then coming to verify whether that has anything to do with the inflammation part of the disease. You've done it in human, but then when it comes to verifying it, you need to step down. One thing I want you to have at the back of your mind whilst we step into the results from this part of the project is that the human endometrium is not the same as the mice endometrium. And various studies have found that usually when you induce endometriosis in a mouse and you don't even treat it, you don't do anything to it, it is able to resolve by itself. And that is one of the reasons why mice models or mouse models of endometriosis have not been able to successfully replicate the way the disease is in humans. The next thing that they did was that they took some mouse, first established the infection of the Fusobacterium nucleatum, and then take some uterine tissue from BAPC mouse, ground it, and intraperitoneally inject it into the mouse that had already had Fusobacterium, and then wait for the endometriotic lesion to establish. In the mouse where there is Fusobacterium infection, they found more lesions compared to the ones that did not, and then treated two of the groups with metronidazole and chloramphenicol, which are two antibiotics that the Fusobacterium is susceptible to, and so is able to eradicate them by introducing it transvaginally for 5 days and look for possible reduction in the lesion. So over here, I think, in my opinion, where this particular study controls fall short is having in mind that when you introduce foreign endometrial tissue into the peritoneum of mice and you even leave them, they are able to resolve and get healthy. There should have been another control where you give them an anti-inflammatory drug to see if it is going to give you the same outcome. Then your claim that treating the Fusobacterium with an antibiotic is what actually resolved it. But there was no such. There was just the metronidazole and then the chloramphenicol and then the ones that were not treated, nothing else. So if any intervention at all could give you the same outcome that you saw with the metronidazole and the chloramphenicol, then you know something else is happening and not just the eradication of the Fusobacterium. But the important thing that I want you to pay attention to is some of the things that the researchers said themselves in the paper when they were talking about the limitation of the study. So you could see that they said that this study has some limitations that need to be considered when interpreting the results. And the first limitation that they spoke about is that the study lacks direct evidence supporting the hypothesis that the presence of Fusobacterium in the endometrium promotes endometriosis after retrograde menstruation. Like I mentioned earlier on, that for instance, if you take just 4 endometriosis patients, particularly with ovarian endometrioma and other patients who do not have, you screen them, that number alone is not enough to make any significant conclusion given the complexity of endometriosis and the heterogeneity of the disease. So one, the sample size was very small. They also say that considering that many studies demonstrating mechanisms responsible for endometriosis, further precise studies are needed. So they are also acknowledge that there are other studies that are mentioning several other processes as a cause for endometriosis, and then some of them still have questions that still needs to be answered, especially with retrograde menstruation, that is the most widely accepted. Still, we don't know why you have retrograde menstruation occurring in about 90% of women in their reproductive years, but then just about 10-15% of them get endometriosis. So what is happening to the remaining 90-85%? Why are they not getting it? If we don't have answers for that, we can definitively say that, yeah, retrograde menstruation causes endometriosis. And the researchers pointed it out that further investigation needs to be done to ground that claim in truth. But it's good to mention so that it prompts other projects, so that it prompts the need for larger study to look into it to see whether it is possible that treating the Fusobacterium could actually be an effective management option for endometriosis. And then the second thing they spoke about is that the clinical studies are required to ascertain whether treatment with antibiotics against Fusobacterium represent a bona fide effective treatment for patients with endometriosis. Like I said, the human endometrium is not the same as the mite or rat endometrium. It's not even the same as a baboon's endometrium, which is the most closest genetically animal to humans. And so anything that you look for, you find in the mice, it's not something that you can clinically translate back into humans. While studies of this are important to prime the need for large-scale studies of its kind to verify or otherwise the findings that were reported, in itself alone is not enough to ground the claim that Fusobacterium infection causes endometriosis, and or the eradication of Fusobacterium infection is enough treatment protocol for the management of endometriosis. The evidence of this lies in the lack of any of its kind or a mention in any of the globally accepted endometriosis management guidelines. If you find the page and the content here insightful, share it with others in your network that you think will find it beneficial. Give us a like if you found the video interesting. Comment what your thoughts and your perspective are about the video. This helps the YouTube algorithm to determine that what we are doing here is important and then recommend it to more people so that the page can grow. Endometriosis Voices, listening to lived experiences, learning from science.